Hello, my name is Melissa Tand and I am majoring in Civil and Environmental Engineering. My mentor is Dr. Thinesh, and the title of my project is Studying the Extracellular Polymeric Substances of Galdieria sulphuraria as Flocculation Aid for Improving Algal Harvesting Efficiency. Biodiesel has been produced for the past 50 to 60 years, and one of the most promising sources of biodiesel come from microalgae. Because of their low biomass densities, harvesting microalgal biomass consumes a significant amount of energy and has proven to be a significant economic challenge. Algae are known to excrete extracellular polymeric substances, or EPS, into their surrounding environment. EPS contains many biomolecules, and one application of these biomolecules lies in the flocculation process for algal biomass harvesting. Flocculation is a relatively low energy consuming process, and by using EPS as a flocculation aid, the cost and energy demand for algal harvesting could be decreased. In this study, the EPS of Galdieria sulphuraria was used to conduct flocculation experiments to assess the feasibility of using EPS to improve algal harvesting efficiency. Currently, this project is still ongoing. So far, we have already extracted the EPS of Galdieria sulphuraria. Unfortunately, due to the limited time and a limited amount of large reactors inside the lab, only 3 flocculation experiments were conducted. Each experiment requires a large amount of culture, and 2 months were spent growing and maintaining these cultures. In the following slides, there are results from the 3 experiments that were conducted. Experiment 1 was conducted using 50 milliliter reactors, and Figure 3 shows the experiment setup that was used. The optical density, or OD, was measured at 750 nanometers. The OD value was used to measure the algal biomass concentration, and 1 OD is approximately equal to 0.5 grams per liter of algal biomass. After 10 minutes, 30 minutes, 1 hour, and 2 hours, the OD value was taken from the midpoint of each reactor. 3 conditions were set for this experiment. The first condition was the control with no EPS added. The second condition was 1 milliliter per liter of EPS added, and the third condition was 2 milliliters per liter of EPS added. This experiment was conducted to see if changing the amount of EPS would affect the harvesting efficiency. From the results of Experiment 1, we can see that the OD values of the reactors with the added EPS had a significant decrease after 10 minutes. A lower OD indicates that there is a higher harvesting efficiency. After 10 minutes, the control had a 32% decrease in the OD value, whereas the other 2 conditions with the added EPS had over a 70% decrease. However, after 30 minutes, all the reactors showed around the same OD. Experiment 2 was conducted using 1-liter reactors with 700 milliliters of working volume. 2 conditions were set for this experiment. The first condition was the control with no EPS, and the second condition was 1 milliliter per liter of EPS added. In this experiment, the OD values were also taken after 10 minutes, 30 minutes, 1 hour, and 2 hours. Figure 6 shows the results from Experiment 2. The first graph shows the changes in OD, while the second graph shows the changes in harvesting efficiency. At all times up to 2 hours, the reactors with the added EPS had lower OD values than the control reactors without EPS. This means that the reactors with the added EPS had higher harvesting efficiency within the first 2 hours. Experiment 3 was conducted using 50 milliliter reactors. This experiment was set to see the effects of using more EPS. Compared to using 1 milliliter per liter of EPS in Experiment 1, 2.5 milliliters per liter of EPS was added to Experiment 3. In addition to adding more EPS, this experiment was also conducted using 2 different initial algal biomass concentrations, or in other words, different initial OD values. 4 conditions were set for this experiment. The first condition was Control A with an initial OD of 0.6 with no EPS. and the second condition was an initial OD of 0.6 with 2.5 milliliters per liter of EPS added. The third condition was control B with an initial OD of 1.2 with no EPS. And the fourth condition was an initial OD of 1.2 with 2.5 milliliters per liter of EPS added. For this experiment, the OD values were taken after 10 minutes, 30 minutes, and 1 hour. Figure 7 shows the results from experiment 3, and we can see that the reactors with the added EPS had lower OD values than their respective controls. Due to the limited time and unavailability of large reactors, only a limited number of experiments were able to be conducted. The addition of EPS seemed to improve short-term harvesting efficiency up to 2 hours. Specifically, the harvesting efficiency was observed to be higher within the first 30 minutes. With that being said, more detailed experimentations are needed to make a conclusive argument that the addition of EPS improves algal harvesting efficiency. Lastly, I would like to thank the Office of Undergraduate Research and the Department of Civil and Environmental Engineering for funding this project. This project was also a team effort with the help of Simone, Ari, and Dr. Thinesh. I would also like to give a special thanks to Dr. Barim for everything he's done. Thank you for listening to my presentation.